All right, so you ready to stab yourself? So the first thing we're going to do is we're going to test for blood sugar. The reason we're going to do that is because we're going to get a little bit of a glimpse into how blood sugar can change over time. Okay, because I am going to, I maybe haven't posted Monday's recordings yet. I will do that here in a minute, maybe, hopefully. I'm glad you said that because I did forget about that part. Uh, So what we'll do is we will first go ahead and look at our blood sugar now, because this is also going to be a little bit of review of last time. Okay. Because what's going to happen is we're going to see what our kind of resting blood sugar is right now. I don't know if you've eaten or not. If you had if you have, you probably expect that your blood sugar is already going to be somewhat elevated because you had some food. But we're also probably have some insulin in our system trying to bring that stuff down. But then what we'll do is I am going to put some sugar in your system in the form of chocolate, uh, so that we can really cause those numbers to spike. Uh, and then when I like y'all will be doing that while I'm talking, then hopefully we can check it a little later to see what has happened to our numbers. Okay. And then when we check in a little while, we'll also go ahead and do another check with the blood and where we will actually look at the ABO blood typing for your blood. Okay. Sound good? All right. Now, to do this, we do have a couple of things that we need to do before we just willy-nilly stab ourselves. The first thing that we're going to be needing to do is going to clean our skin with one of these alcohol pads. So you're going to want to pick one of your kind of side fingers out here. So don't pick your thumb, don't pick your index finger because we touch too much stuff with those. Also, we're not going to be stabbing ourselves on the pad of our fingers, so like on the underside. One is because it's incredibly sensitive, and two is because we touch everything there. So you're going to make something hurt far more than it does needs to. So what we're going to do is we're going to kind of pick the side of one of our fingers. Okay. Uh, so just kind of go ahead and pick your poison there, and then we're going to clean it with one of these alcohol swabs. These just tear open. Mhm.
It's like, are we doing it the way, okay, because I'm a medical assistant.
Okay.
So we use little lancets right on the side, and then the first thing is to squeeze it a little and then wipe off the first drop. Are we doing that?
That is best practices.
Yes.
Yes. You do not have to do that if you don't want to. Like if you're not a big bleeder, so that you don't think you're going to be able to generate that much blood, you cannot do that. But yes, that is best practice.
I've had to like, because we did this at high school, right? So nobody really cared like if we did it right or not. So everybody had like 2 pricks per finger.
Yep. Now we're also going to be using, so before we stab ourselves, we're just kind of getting yourself prepared. We've also got our little machines here. It's a good idea to go ahead and get these turned on and ready to go. So the way we're going to load these is going to be, or what we're going to use, we're going to use one of these little chips down here for the people online to see. I'll let y'all kind of take a look there. We've got on this end right here, this little computer chip bit. You're going to face the computer chip looking side up, and you're going to go ahead and place it into the reader, and that should cause the reader to turn on. If not, you're going to push that little black button. Now, when you take that piece and you push it all the way in, you're going to see this little flashing blood drop here. That tells you that it's ready to read. Okay. So once you've done that, and you have some blood, you're just going to take that drop of blood and you're going to press it against that little chip right there. Okay, just press it up against it like this. You're not trying to put the blood on top of it because this is what we call a capillary bed. Okay, so this is a really thin gap that will cause the blood to shoot into the gap, and it's going to go up. And as long as it goes up to that green line, it will give you a number. Okay, it's ready for the blood when you see that little flashing thing. So what I like to do at this stage is go ahead and have your chip in there and close. Go ahead and stab yourself. Get that first drop of blood wiped off so it's nice and clean, uh, so that our results are at least more consistent. And then push it the rest of the way in. Get it ready for the drop of blood. Does that make sense? All right. So, for the entertainment of all of the online people, I will go ahead and stab myself on the internet for your amusement. have already got marks from earlier, so I need to try and not stack those up because that's a good way to make this far more painful. All right, now, so these are little lancelets. So the way these work, these basically have a little spring in here. So all you're going to do is you're going to take that little red piece, you're going to kind of press it where you want it to go, and then just look away and push it down. Okay, and you should hear the little plunge, and then it will stab you. And then, oh look, we have blood. Blood. Okay. Oh, you have a glucometer, but you can't find it? Well, don't stab yourself until you find the glucometer. So the little arrow means that it probably needs to be pushed in further. All your supplies are in the same. Man, Jennifer, you're just messing it up today. I'm joking. You're fine. You'll be okay.
Do I need to stab myself again?
No,
I
just
think
squeeze
that's-
split out. Just squeeze. Just squeeze. No, down here. Like start at the base and just squeeze all. Yeah, there we go. Here. Sit down.
Oh, okay.
And when you're done with that, this one arrow's in. Oh, gestational diabetes. A surefire way of making a pregnant woman who's already likely to be pissed off more often than not, making her even more angry when she has to wait 15 extra minutes at every single one of her OBGYN appointments. Ask me how I know. My poor wife. With both of our babies, neither one did she actually have gestational diabetes, but both of them, from the very first appointment, were considered at risk. So she got tested every single time. It was lovely. Did we get a number?
113.
113. Let's see what we got here. There's my coffee. Oh, luckily yours was just for 2 weeks. That's good. All right, so I'm gonna go ahead and write some numbers down. Mine is 128. I heard a 113. What's our other one? 124. We're actually all right there in the same ballpark. Sweet. All right, so you can go ahead and use your alcohol swab and get that cleaned back off. Your little bit thing right here, you can just go right here into the sharps container, as can your lancets. And then I've got some bandages if you need them.
Also, this one's bent, so do you want to put this one away?
Yeah, you can go ahead and.
That way. My finger won't stop moving.
Okay, everybody. No, I made the mistake last semester of I lanced within half a millimeter of the same exact spot from the first class to the second class. I'm normally good about like bouncing around, but they were super close together, so my finger was already like swollen and mad about it. So that second shot hurt. Like I'm normally like, oh, it's fine. Like it's a prick. Like I grew up fishing. Like I still fish all the time. Like my hands are always scratched up or cut from something. But that was like one of the few times being like, son of a bitch, that hurts. All right. So now with all that being said, we now have some numbers up here. Now what we're going to do is I'm going to bribe you for having come to class and being willing to stab yourself by giving you candy containing some amount of sugar. You don't have to eat it all. You can eat however much sounds good.
I quit.
Exactly. It's like, I'm not messing up my diet. I'm doing it because I was told to.
Yeah.
Now, in my first class, I did not eat any candy. I had breakfast before the class and had some sugar in the form of coffee, but I did not eat any candy during the class. My blood sugar fell during the class because I'm sitting up here talking. I'm actively burning calories. Y'all may think I'm being lazy, but I'm actually doing stuff. Okay. Now, everybody else, it went up because they ate some candy. So now we're going to see what happens when I eat some candy. Okay. Now, let's go ahead and take a peek. Let's now talk about the other stuff that we see other than just blood sugar. So, blood. Blood is a connective tissue. It's a connective tissue because we have a bunch of cells and then we have those cells suspended in a matrix. Okay. In this case, the matrix is liquid. Our blood plasma is the actual matrix that the blood cells are suspended in. Okay. Thus, blood is a connective tissue. Now, what do we use blood for? Transporting nutrients, transporting gases. It's our primary form of transport. Hormones, like we talked about last time, waste. We transport stuff in it. It's basically a highway. If we need it to go from one place to another, we pump it into the blood. Makes sense. Now, we of course know that our heart is doing the pumping, and we're pumping that blood through our arteries, our veins, and our capillaries. Importantly, capillaries are going to be the kind of functional unit. These things are very, very small. And because they're very small, this is actually where the gas exchange and nutrient exchange and all that kind of stuff is going to occur. Okay. Now, our blood is going to be composed of two main things. We have the cells, and then we have the liquid portion, the fluid, okay, or the plasma. The cells, we have three main types. We have erythrocytes or red blood cells, we have white blood cells, and then we have platelets. Sometimes you'll hear platelets called thrombocytes. I disagree with that term for many reasons, namely because cyte means cell. Thrombocytes are not and have never been a true cell. They do not have a nucleus. Okay. So because of that, I kind of disagree with calling them that. Now, it's not that they have to have a nucleus to be called a cell, but they don't do any of the other stuff that normal cells do anyway. They're just a little fragment of a cell that helps us prevent blood loss, which is very important. Okay, stopping ourselves from bleeding when we stab ourselves with a little lancet is important. Okay, and that's what our platelets are there to try and do. But our red blood cells are the reason, well, that our blood is red. Okay, so that is a very important thing. That is also going to be what we're going to be using for oxygen and some of our CO2 transport. Is going to come from our red blood cells. That's why we have so many of them. Okay, we have such a high need for oxygen that we have to have anywhere from 45 to 55% of our blood volume be responsible for carrying oxygen. That's a lot. Okay. Then we have white blood cells. White blood cells are cool because these are actually going to help us defend against pathogens. So defend against the bad stuff. Well, then go ahead and test it. You'll see, like, test it now and then test it in an hour or so, or 30 or 45 minutes when we retest, just to see if it goes down. Although it might go up. It just kind of depends. All right. Now, what are we also using blood for in addition to transporting nutrients? So one of the big functions of our blood transport is actually going to be body temperature regulation. We can use movement of blood to adjust temperature in places. That's because blood is around 1 degree Celsius warmer than the rest of our body. Okay. So because the blood is so warm, if we move blood from one place to another, we can then take that warm blood and then warm up that other location by simply pumping more blood to it. And you've seen this happen. You've probably seen this happen today. When you got out of your car this morning and it was cold outside, like it was 50 degrees, and you walked from your car somewhere else, your face got flushed. Why? And your hands probably turned a little red because it's cold outside. Your hands and your face are exposed. Thus, your hands and your face, those tissues get cold. So what do? We warm the blood up in our muscles as we're walking around, and then we pump that warm blood into those cold tissues. We send more blood to them, so those tissues turn red and they get warm because of the extra blood that we're sending there. Okay, so we can use that to kind of move heat around. Oh, you're cooking and making up. Oh yeah, then no, it'll probably go down. But that's cool. That's still a good hypothesis that we can test. So by all means, test it. So stab yourself with a tiny lancet. You'll be okay. Or a fish hook, whatever you have lying around. Don't do that with a fish hook. One semester, I literally had some fishing stuff in my pockets. I fish a lot, so I usually have some amount of fishing. You're just hanging out in my truck. So I brought one in here just to show people that it's not that bad. Like stabbed myself with a fish hook and used that to test my blood. Because people are sometimes scared of the lancets where the needles are so small you literally can't see them through the thing. It's like that is the world's tiniest needle. It's not going to hurt that bad. So like I pull out this big like 4/0 fish hook. Dink. Here, it's fine. pH is also going to be very important. Blood. Anyone know what blood's pH is?
I think I know, but I don't know.
That's fair. It's 7.4. Okay. We maintain blood's pH in a range of 7.35 and 7.45. That is an incredibly narrow range. Okay. Incredibly narrow. That is the difference of 10 hydrogen ions in 10 million particles of water. That is incredibly small. 10 in a scale of 10 million. Okay, we're not allowed to vary more than 10. How do we keep it in such a narrow range? Well, we use buffers, right? So we use the bicarbonate buffer system here. Well, the bicarbonate buffer system is so good at maintaining pH that we can actually use the blood to buffer other tissues. Most of our tissues do not have to have such or quite as narrow of a band of pH. But if their pH gets a little too elevated or a little too low, we can push their excess acids or their excess bases into the blood to allow the blood's buffer system to bring it back down to normal. Okay. So we can kind of use the blood's pH buffering to help buffer the rest of the body's pH. Okay. That's also part of why it can be such a big deal to mess with the blood's pH so much, is because if it gets messed up, it messes up all of the other pHs too. Okay. Now, we of course are going to use it for fluid balance as well. We drink water, we take in water with our food and stuff like that. We're going to store that water in the blood. If we need to up or down regulate water from our urine, we're going to be up and down regulating the water in the blood, which is also going to be messing with things like pH. Now, we're all going to typically have, or like an average sized average person is going to have around 5 liters of blood. Okay. However, that is an average sized average person. By that, I mean, on average between a male and a female, averaging those sizes and take an average of each of those. Okay. So this is an average. This is going to vary a lot, depending on how big you are. Shaquille O'Neal is 7'4", 350 pounds. If that man had 4 liters of blood in him, he would be dead. Okay. His body would simply not function with that little blood. He has to have more. The bigger you are, the more blood volume you're going to have. You're a lab tech, so you've seen people have blood drawn. Bigger people, when you hit them with one of those things, they will fill up that vial 2 or 3 times faster than someone that is smaller. That's not a joke. Like if you take some like little tiny like old grandma who's 5'2", $110 maybe, On a good day, she'll take much longer to fill up than someone like me. Like it's hilarious how fast I can fill one of those vials. Like they hit it and it's filled. Like you go. It's also fun seeing the phlebotomist like, oh, I can see your veins like sitting down and you're not even prepped. Like my job is easy. uh, but that kind of stuff matters. The size of the needle is going to matter on how much blood they're making or how much blood they're going to have. It also means that those larger folks, it's going to be easier for them to donate a liter of blood and not have any ill effects because, well, they have enough extra where it's not that big of a deal. Like I can genuinely like donate a liter of blood and go run after and not feel any ill effects just because I have enough blood volume behind me. Not the case if you're smaller. Now, we've talked about that. All right, so now let's go ahead and look at blood. So up here on the screen, you can see this fun little test that we call a hematocrit, okay? Or a whole blood separation, okay? If you were to take a larger vial of blood or to fill up a big capillary tube, we can then spin those down in a centrifuge and we're going to separate the blood into its components, okay? One of the first things that you're going to see is this big chunk of red down here. So the proportion of this overall column that is red blood cells is a number that we call the hematocrit, okay? And this is basically just a number that says, okay, what proportion of your whole blood is red blood cells? This is typically going to be anywhere from 40% to around 55%. That's a big range, and we're going to see kind of that range be a little bit different for males than it is in females, and we'll talk about why later. But on average, we'll just say kind of around 45%, okay? Now, sitting on top of those red cells, so the reason they end up down here is because when you spin something really fast, bigger, heavier things are going to move towards the bottom, okay? Our red blood cells are incredibly dense, so they all get pushed down here. The next little line is going to be less than 1% of our overall volume, but it's going to be this kind of white or silver layer. Because of this, we're going to call this the buffy coat. This buffy coat is going to be composed of white blood cells and platelets. Okay, so our leukocytes and our platelets are going to make up our buffy coat. And like I said, this is typically going to be around 1% or less. And then we've got the rest of this, which is the liquid portion of the blood, which is our blood plasma. Now, the vast majority of our plasma is going to be water, okay? Over 90%, so something like 92%, 93% of this is going to be water. The other 7% is going to be proteins, okay? Primarily, this is going to be albumin. So you've actually seen the protein albumin before if you've ever cracked an egg open. Okay, the white part of an egg is albumin protein. It's very similar to the albumin proteins that we actually have in our blood. Okay, and what we use those albumin proteins in our blood for is to kind of make the blood plasma watery. Okay, albumin is a big pull on water. It's what we call a colloid protein. Okay, so water sticks to it. It kind of calls water to it. So if we have enough albumin, we can call a bunch of water and stuff to it so we can maintain some amount of water in the blood. Okay. We've also got things like fibrinogen and some other regulatory proteins in here as well. And then finally, we have all of the other solutes that we're going to find in our blood, whether it's some of our dissolved CO2 or electrolytes or sugars in this case. So y'all just had some sugar get injected into your blood, or it should be there by now. So we're getting all that stuff into the blood, and that's going to make up some small portion of that blood plasma as well, okay? Now, typically that hematocrit is going to vary, of course, between folks. And this is just nothing more, nothing less than the percentage of red blood cells. In males, it's going to be anywhere from 42% to 56%, okay? The higher the number, the more viscous the blood is going to be. In females, this is going to be anywhere from 38 to 46. Notice that it starts lower and still in like the high end for females tends to be on just barely overlapping with the low end of males. Okay. Now, one of the big reasons that causes this is testosterone. Okay. Increased levels of testosterone are going to lead to increased levels of erythropoietin. Okay, erythropoietin is a protein that is going to cause us to make more red blood cells. Okay, so having more testosterone means more erythropoietin, which means we're going to have more red blood cells. Okay, which means we have more oxygen carrying ability in the blood. That also means we have more tissue regeneration ability in the blood and stuff like that. So males will tend to recover from things like athletic activity and stuff faster. Now, if we look at a blood smear, so instead of just taking your blood and putting it in a vial, instead we spread it onto a microscope slide and then we throw some stain on it. The stains that we use for blood are going to stain our white blood cell nuclei purple, and it's also going to stain the granules inside those white blood cells particular colors depending on what types of leukocytes they are, so that we can use that to look at not just the number of red blood cells, not just to see them, but now we can start to count these. One of the tests that's very commonly ordered is going to be a whole blood count. Okay. Uh, a C or a complete blood count, so a CBC. Okay. What they're going to be doing in those cases is that they're not just going to be looking at the number of erythrocytes, they're also going to be counting our white blood cells and then counting the types of white blood cells within that. Okay, so we can say we have this number of leukocytes, we have this number of neutrophils, we have this number of basophils, this number of eosinophils, because different types of infections are going to cause those numbers to be elevated in very particular ways. So we can kind of monitor for particular types of infection that way. So we talked about some of that already. All right. Now let's talk about making blood. So I mentioned a little bit ago. So the process of making blood is what we call hematopoiesis. Okay, this happens in the red bone marrow of our axial skeleton, as well as in the head of the humerus and the head of the femur. That's it. So we're talking very, very much axial, at least in adults, that this process is occurring. Okay, it is relying on a particular cell called the hemocytoblast. This is the primary stem cell. This stem cell, so a hemocytoblast, can turn into any single type of blood cell. Okay, it can turn into a red blood cell. It can turn into a white blood cell. It can turn into any different white blood cell. Okay, and that's all going to happen based off of particular chemical triggers. Okay, if we expose that hemocytoblast to a particular chemical trigger, we can have it turn into very particular things. We can push it into one direction or another. Okay. Now, one of those directions that we might want to push it into is what we call erythropoiesis. Okay, so erythropoiesis is going to be right here. So erythropoiesis is making red blood cells. Okay, now the first trigger up here, we can form one of two things. We can either form what we call the lymphoid line, so we form a lymphoid stem cell, or we form a myeloid stem cell. A lymphoid stem cell can only become types of lymphocytes. We only have three options here. We have B, T, or B lymphocytes, T lymphocytes, and natural killer cells. These things are awesome. Okay. Our other option are our myeloid stem cells. Myeloid stem cells can turn into our platelet producing cells. They can turn into our granulocytes or monocytes. So they can turn into many of our different types of white blood cells, or they can turn into red blood cells. The way that we turn them into red blood cells is with the hormone erythropoietin. Okay. So erythropoietin is super fun. Okay. Erythropoietin, like I said, is going to cause us to make red blood cells. It's going to be secreted by our kidneys, particularly by our kidneys. A tiny little bit gets made by our liver. If your kidneys realize that we're starting to run a little bit low on blood, that can trigger them to produce more erythropoietin, or just simply having higher testosterone levels will cause them to produce more erythropoietin. When that happens, well, if we increase the amount of this erythropoietin that we have right here, more of our myeloid stem cells are going to get turned into erythrocytes. So we're going to produce far more erythrocytes, which means we're able to get more oxygen into our tissues more often, which means we're going to be quite good at things that require lots of endurance. Okay. A good example of this would be running or biking, like a big endurance event like the Tour de France. Probably the single toughest endurance event that we have in sports. Okay, it's a 2.5 week ordeal where you get 2 days off in the entire 2.5 weeks, and you're biking anywhere from 100 on a short day, maybe 50 on a sprint day, but anywhere from 100 to 500 kilometers in a day. Much of that up hills because we're in the Alps in France. One of the toughest a single day of this could be considered one of the toughest athletic events in the world. And they do it for 2.5 weeks because they're batshit crazy. Okay? Now, if you know that you're going to be doing that, you're stressing your muscles and you're stressing your mind to an absurd degree. So what do you want to do? You want to make that as easy as you possibly can. Well, the idea that all of a sudden you can make your muscles have more endurance so you can work them harder on your exercise days, but then more importantly, or just as importantly, while you're asleep, they're getting more oxygen and stuff like that to recover faster. Kind of a no-brainer that you would do that. The problem is that the World Doping Agency knows how to test for testosterone. So steroids you can't do. Also, steroids when you're a biker and you can't weigh more than 140 pounds when you're soaking wet, just because you can't, because it's more mass that you have to move around, not good. So what do? You just want the blood aspect, right? So instead of doping with the testosterone, which can be tested for and also kind of hurts you in some ways, instead you just want the other benefits. So you inject yourself with EPO, erythropoietin, so that you do more of this. So that you make more red blood cells so that you can recover faster. You do this, and if you train really, really hard, because now you can train incredibly hard because you recover better, you turn into the best biker in the world and you win 5 or 6 of these things in a row until someone writes a book that says, hey, by the way, we were all cheating. And that's what happened to Lance Armstrong. Okay. Lance Armstrong won 5 or 6 straight Tour de France's. He was considered the greatest athlete of our time. Keep in mind, Michael Jordan exists. And there were some people calling Lance Armstrong one of the greatest athletes of our time, if not the greatest athlete of our time. That's how hard this event is considered. But later on finds out he cheated and he was cheating with EPO. Granted, basically everybody else was then too. Okay, the only way to be competitive in the bike sports back then was to do that. If you did not cheat, you were not competitive. So there is not considered a winner for any of those events anymore. They basically have wiped all of the leaders out of those. But yeah, it's all because they took this stuff, so they got a whole lot more red blood cells. However, can you think of any problems that we might have if you have too many red blood cells? Didn't I say that it's the number of red blood cells that make your blood as viscous as it is? If you add a whole bunch more and you start looking at 65 or 70% even on a hematocrit, you can get clots. But also, your blood isn't really liquid at this point. It's like half done jello. It's goopy. It's so thick that it's literally goopy. Like it will, like you can invert a cup and it won't spill out. That's not a joke. Yeah, kind of like Dairy Queen. There are videos on YouTube of like bodybuilders that have like put some of their blood in a cup and you can literally do this. Their blood is so thick. And that's because this is one of the key benefits of injecting yourself with stupid amounts of testosterone, is because you're getting all of this benefit to your blood. All of this benefit to your blood is going to be a big benefit in your training ability and your recoverability and stuff like that. And in the case of an actual bike event, something like that as well. So that's why they do it. But once again, this can put a heck of a lot of stress on your heart because it's having to pump this goop through your system instead of liquid blood. It's also very difficult on your capillaries. It can lead to clots and all kinds of fun other issues. So it's definitely not good for you to do that, especially over a long period of time. But a lot of people do still do it. All right. Now, the last one that we're going to talk about is platelet formation. That's because this is my second favorite cell in the body. My first favorite is the osteoclast. You'll notice this looks very similar. It is also another like monster squid looking creature. big, huge cell, thus megakaryocyte. It's got these little tentacle-like appendages that reach down into your blood vessels. Okay, so in this case, our blood is flowing from left to right. Okay, so our blood's flowing from left to right. So as the blood is passing over these cellular extensions down here into the blood vessel, well, what happens if you say take a water hose and spray it on a soft plant? It'll start to shred off little bits of the plant, right? Well, the same thing is going to happen to our megakaryocytes' tentacles. Those tentacles are sticking down there in the blood, so as the blood is passing over them, it's just going to cause little bits of those tentacles to just shear off. So it's just constantly pushing new tentacle down in there so that we're constantly producing more platelets. And these platelets are going to help us have things like clotting factors and fibrin and all of the other kinds of things that we need to help the blood clot, in addition to having just the platelets themselves, which help in clotting. But they're super cool looking. I mean, look at that thing. Like, that's just freaking cool. It's got little feet. All right. Now, erythrocytes, red blood cells. Red blood cells also have a very particular shape. We have this well-known kind of characteristic biconcave shape. Now, one thing to keep in mind with this cell is that this cell is not rigid. Okay. You can think about this in very much the same way that you think about an inner tube on a lake. An inner tube going around the lake is also not rigid. It bends, it flexes. Okay. You can put it under stress and it will bend. The same thing is true of these erythrocytes. You hit them with a bunch of blood pressure, you hit them with a bunch of fluid, it will bend. Okay. So this flexibility is going to be important for them because they're going through capillary beds that are curvy, bendy, twisty. Okay. Now, this biconcave shape also makes them very energetically efficient moving through the fluid, but it also gives them a lot of efficiency in terms of surface area to volume. Okay. We have a lot of surface area, we don't have a lot of volume, which means diffusion has enough time to get oxygen into the hemoglobin right here in the center, as well as out. Why? Because this distance is not very far. Why is the distance not very far? Because there's a divot in the center of the cell. Okay. It's quite literally the same reason that donuts have a hole in them. Donuts have a hole in them so that you can cook the middle of the donut. Okay. Heat has a chance to get in and actually cook the dough in the center part. If you don't have a hole in a donut, the inside is raw most of the time. If the outside is going to be nice and crispy and stuff like that, the inside of it would still be raw, which is not ideal. Okay, but if you poke a hole in it, it all cooks evenly. So the same thing is happening here. By having this divot, we now can get oxygen and CO2 to the inside of these cells very efficiently. But this shape also lets us do something else. This shape lets us stack the cells. Okay, so I like to think about the blood vessels as highways. Okay, so a regular artery or vein is basically going to be like a highway where we've got multiple cars all moving in the same direction. Your capillary beds, though, are going to be like a toll booth in the middle of your highway. So like say you're going to DFW and you have to go through one of those old school toll booths. Well, what happens? Do they just take it like, do you go through the toll booth 2 lanes at a time the same way you would if you were driving? No, they branch it out into like 16 or 20 lanes, right? So that we get more surface area. Okay. So that we can move more cars through it more quickly. So we fan things out. The same thing is going to be happening with our capillary beds. We're going to open them up into a bunch of big lanes. Okay. But these lanes are also very small. A capillary bed is just a little bit bigger than a red blood cell. Okay. So the red blood cells have to pass through one at a time. So what's happening is these cells are quite literally stacking up side to side to side to side to side to side to side. They slow down to go through the toll booth, just like you have to slow down and go through the toll booth. Why do you think we need to slow them down? Exactly. By slowing them down, this gives us more time to transfer oxygen and wastes and stuff like that. This gives us enough time that we can get oxygen to bind to them or to get oxygen that is bound out and into the tissues. Without the time, that doesn't happen. So if the blood moves through the capillary beds too fast, so if the capillaries are too big, we don't have efficient transfer of nutrients or oxygen in and out. Does that make sense? All because their shape allows us to make them stack, which is very nifty. Now, one of the other things that's going to make those cells maintain that shape is that they are all going to be filled with hemoglobin. Okay, hemoglobin is awesome. Okay, This is what hemoglobin looks like. We've got 2 alpha chains and 2 beta chains. In the alpha chains, we have 2 heme molecules. Okay, these heme molecules look like this, and they're going to be associated with an iron. That's this iron in the middle. There are so many hemoglobin molecules in a single red blood cell that if you put a red blood cell on or red blood in your mouth, you will taste the iron. There's so much hemoglobin in those cells that you can literally taste the iron that is in it. Okay, that metallic tang to the taste of blood is because of the iron and hemoglobin. But this is what hemoglobin is supposed to look like. Okay, and if hemoglobin looks the way it's supposed to look, your cells will look like this. However, we have a problem. One very common issue is to take that hemoglobin and instead of having it be in this kind of square shape, is that we reorient some of these proteins. So and instead, they look like this. The problem is that these ends are sticky, okay? So that if we take another hemoglobin that is this shape, it'll stick right here. And then another one will stick right here, and right here, and right here. There are as many as 100 million or 300 million hemoglobin in a single red blood cell. That's a big number. And if we start to make a whole bunch of them into these chains, something fun happens. Instead of the cell looking like our nice little red inner tube, when we've got our lovely hemoglobin doing what it's supposed to do, instead, when it does this, which typically happens in a high temperature, high pressure situation. So high blood pressure when someone is being very, very active or something like that. So under a stressful scenario, all of a sudden, this will happen. You can't see what I'm drawing. It's not a great drawing anyway. It's fine. There we go. It will make it look instead like this. And it will cause our cells to sickle. This is called sickle cell anemia or sickle cell trait. Okay. This is all caused by a single base pair change in that protein. In one base pair change, it's a C to a G in one of those subunits. That subunit contains something like 15 or 1600 bases. One change is enough to make your blood go from looking like this to looking like this. Now, unfortunately, this is also oftentimes too long to fit through a blood capillary. This is a capillary. If it goes in this way, all of a sudden blood stops moving through the capillary. So exchange is no longer occurring. And blood is packing up down here. And then eventually, if it happens in enough of those capillary beds in that particular region, you'll start to bleed around it. It'll literally build up so much pressure that it breaks out. And these people will bleed from underneath their fingernails and out of their eyes. This is usually one of the last things that happens to them, though, because oftentimes that's enough to cause death because they're bleeding in other places that we can't see. This has killed teenagers on sports fields. This is also one of the reasons why they do genetic testing now in a lot of our, like a lot of school physicals and stuff like that for sports will include genetic testing now, specifically looking for that. Because we don't want to have a 16-year-old kid die on a football field due to a sickle cell attack. That is scary stuff, okay? But we live in an incredibly cool time because we are now alive in a time to where that one base pair change is fixable. Unlike any other time in history where all we can really do is treat the effects afterwards and try and prevent it, now we can give you a drug that will change that letter from being wrong to being right.
That is pretty freaking amazing. One of the first gene editing things that was done in humans, like therapeutically, was fixing sickle cell anemia. And now, if you have it, you can literally go to the doctor and start undergoing treatments to fix the genetic problem. That is flipping cool. All right. Let's go back to other stuff. All right. Now, we've talked about those. Here's erythropoietin, red blood cell production. There's a nice little figure talking about it here. I already said all this stuff before, so now we can kind of see it. Oh, one fun thing. Our red blood cells only live about 4 months. Okay, so they only last for about 120 days. Which means we're constantly making new ones and we're constantly getting rid of them. We get rid of so many of them that the two main things that you excrete, your urine and your feces, have their color because of getting rid of red blood cells. We get rid of so many that that's actually what gives them their color. Okay, so the way we get to the brown in our feces is we're going to produce from the hemoglobin. Okay, so from the heme groups, we're going to turn that into bilirubin in the liver. That bilirubin then gets turned into urobilinogen in our small intestine. And that urobilinogen then gets converted by bacteria in our gut into stercobilin, which is brown. And that is the reason our feces is brown. It's because of the components of old, dead, red blood cells. That's pretty fun. The other thing that urobilinogen gets turned into is urobilin. Urobilin gets broken down in the kidneys as a yellow pigment. Colors urine yellow. That's why. So both feces and urine are the color they are because of red blood cells, and particularly because of hemoglobin. I don't know why I find that so entertaining, but I do. All right. Now, what you've all been waiting for, blood typing. We get to stab ourselves again. Yay. All right. So blood typing is fairly simple. Okay. We say that we have a blood type because we have the presence of a particular cell signaling molecule called an antigen on the outsides of our cells. Okay. Our blood types are broken down into two main things. We have the Rh blood type, and then we have the ABO blood typing. Okay. So ABO is going to form the kind of A, B, AB, or O type of blood that you've probably heard about. Okay. And all those types are, they're simply telling you what type of antigen is present on the surface of the cell. Okay. Now, with that being said, I'm going to stop the share again. And hopefully they can see at least a little bit. All right. So for type A blood, we are type A blood because we have the A antigen. So this is a protein that's sticking off the cell that has the A shape. Okay. So if we have the presence of the A antigen on the outside of the cells, we're going to call that type A blood. Now, importantly, this is genetically determined. Okay. There's a spot on your chromosomes that say you are going to produce the A antigen. That same spot on the chromosome can also determine if you produce the B antigen. However, how many chromosome copies do we have of everything? 2. You get 2 copies of almost every gene. Okay? You get 1 copy from mom, 1 copy from dad. Unless it's on the X chromosome, that's a special case. Okay? But if it's not, you get 2 copies. You get 1 from mom, 1 from dad. If you get an A from mom, you will produce this A protein on the outside of your cells. Okay? This protein will just hang out there. Okay? Now, if you produce the A protein, you will also produce the anti-B antibody. Okay? So someone that has type A blood, even if their blood has never been exposed to anything else, will also produce the anti-B antibodies. So just floating around in their blood will be some anti-B antibodies. This means that if you give a patient with A blood, blood with the B antigen on the outside, well, those antibodies will find that antigen and stick to it and cause that blood to clot. Okay. And you will cause a transfusion disorder or blood clotting, and you can stroke your patient out. Okay. Not recommended. Don't do that. Okay. We don't want that. Okay. But so type A antibody or type A blood will have the A antigen and it will produce anti-B antibodies. Type B blood will have the B antigen and it will produce anti-A antibodies. So this person, if you give them A blood, well, they have an antibody that will fight against it and it will cause that blood to clot. But like I said, you get 2 copies of this, which means you could get an A from one parent and a B from another, which means you have type AB blood. Now, if you have type AB blood, what antibodies do you think you have? If you have, like, say you have AB blood and you had anti-B antibodies, well, is there a B on here? Well, that means those antibodies would attack it, right? So we can't have anti-B antibodies because we'd already be dead. Okay. We can't have anti-A antibodies because we would also already be attacking ourselves and be having a problem. Okay. So that means people with AB blood don't have either antibody, which is great because these people can be like, just give me all the blood. It's fine. Doesn't matter. Just give me all the blood. Okay. They will not have a transfusion reaction because they don't have the antibodies to fight against it. Okay. Against either the anti-A or anti-B antibody, which makes them very, very good recipients for blood. Okay. Now, the opposite of this is also true. There is a third option at this locus, and that is to say that we don't produce either the A antigen or the B antigen. Instead, our antigen looks different from either of those. So we call this the O antigen. The O antigen is neither A nor B. What antibodies do you think this one makes, though? A and B. It has anti-A antibodies and anti-B antibodies. If you give this person either blood type, they will have the ability to make it clot. So this person will have a transfusion reaction with any sort of surface antigens that are present on the blood, which is a problem. You have to be very, very careful when you're giving blood to an O patient. Okay. Now, this is all on a single locus. So the ABO locus is in one spot on one chromosome. To further complicate things, we have another spot that determines the presence of another protein on the outsides of the cells. This is what we call the RH factor. Okay. The RH factor is another surface protein. Okay. And we're simply going to call it by presence or absence. Okay. If we have it, so say we have this RH protein, we would call that blood RH positive. Or in this case, we have the type A antigen and we have the RH antigen. So we are A positive. Okay. So if you've ever heard of someone saying, oh, I'm AB negative or AB positive, the positive or negative is simply referring to this RH factor. Okay. So if we have it, we are RH positive. If we don't, we're RH negative. Now, the part where the RH factor is different than the ABO blood typing is that someone that is A negative will not have active anti-RH antibodies moving through their blood unless their blood has already been exposed to the RH antigen. Okay, so someone that does not produce the RH antigen will not have antibodies against it unless their blood has been exposed. Okay, that is different from the anti-A and anti-B that we just kind of innately have. Okay, now what this means is, say we have a female patient and say she's type A blood and she marries someone with type B blood or has a child with someone with type B blood, but say that person with B blood is B positive. Well, now they have an offspring that is A, B, and RH. Okay. Now, during gestation of this first child that has this RH positive factor, nothing will happen because mother's blood and baby's blood are separate.
mother will never get exposed to this Rh antibody, which means she will not be producing or hasn't been exposed to this Rh antibody, so she doesn't have any antibodies to fight against it, so her body will not be trying to fight against the fetus. Okay? Problem is, during childbirth, it's a very traumatic experience for all involved. Okay? That means all of a sudden, mother will get exposed to the Rh factor in the baby's blood during the first childbirth. So baby 1, everything's fine. But baby 2, all of a sudden, mom has anti-Rh antibodies floating around. And now we have a problem. Because if we get pregnant again, all of a sudden we have antibodies that will try and fight against the pregnancy. Now, this was a big enough problem that for a pretty good while, I'm talking like 20 or 30 years, like if you got married in the 50s and 60s and even 70s and 80s, or I guess more like in the 70s and 80s, they would do a blood test as part of you getting your marriage license to tell you whether or not you were going to have this blood typing mismatch. So that basically they would be able to tell you, hey, you shouldn't have more than one kid. Because if you have more than one kid, baby and mom could both end up having problems during the second pregnancy. Like this is bad enough that it could kill either one or both. Okay. However, now we can treat it. Like now we can give some drugs so that we can knock out these antibodies and everybody's fine. But there was a time that this part was not treated.
And it literally did cost like mothers and babies their lives. All in the presence of protein on the outsides of some cells. Ain't that some nonsense? Crazy. But like I said, we can fix it now. Oh, all right. Now I have talked enough that hopefully you have metabolized some sugars. All right, so now what we're going to do is we are going to swab our fingers and we're going to stab ourselves again. And then we're going to check our blood sugar once again. Once you've checked it, come up here and go ahead and write it next, or you can just tell me since there's only 2 of y'all. And I'll write it up here so we can see how those numbers changed. And also, once we've done that, so make sure once again you wipe off the first drop of blood so it's nice and clean, so that we're not contaminating with any sugar and stuff that are on the outsides of our fingers and all that kind of fun stuff. Then we will go ahead and test our blood typing. The way we're going to test our blood typing is we're going to take one of these little trays right here. Oh, I wrote the RH on the wrong set. I'm a terrible human. What we're going to do is the first thing we're going to do is we're going to put a small kind of half a drop of sort of volume of blood into each well. So into the A well, the B well, and the RH well. And then I've got some little vials of antibody here. That's what these are. These are the anti-A, B, and RH serums. And then we're going to put 1 drop of the correct serum onto the spot. So the RH, we're going to put the RH serum on. The B, we'll put the B serum on. The A, we'll put the A serum. And these contain the antibodies. So what we're going to be doing is we're going to be looking for the clots. Okay.
The same finger?
Huh?
Or on a different finger?
If you use the same finger, do it on the other side.
Okay.
If you use a different finger, it doesn't matter. You just don't want to do it on the same side of the same finger because that side will have some local swelling and stuff like that. So it'll be a little bit sensitive. You'll make it very angry. I mean, you can do it if you want, if you don't believe me, but. Yeah, I know you would believe me. It's always like I have seen people like sometimes people don't believe me when I say stuff. It's like, yeah, I'm telling you something for a reason. Like I do enjoy the sound of my voice, but not that much. So online people, that's what we're doing now is we're basically doing the blood typing. So now if you want to do the blood typing virtual lab, that's kind of essentially what you can work on now. But like I said, we're also going to be looking at the blood sugar one too, just because we've had a chance to enjoy some candy. And Jennifer, if you want to use this as an excuse to test yourself again a couple times, just so that you can eat some candy to see if your blood sugar goes up when you do, I think that's a great excuse. Well, you do this one first. Yeah. Do your blood sugar first, and then you can do the tray. Need to find an unwounded spot. 2 classes back to back means that holes everywhere.
Oh, I've got new lanterns for y'all too. Y'all didn't already know. You did.
Yeah, we
One,
had-
two.
Yeah, we had somebody in high school that their little hole wouldn't like close up, so we just used the same thing like over and over and over. Say this 17.
Had a bleeder, huh?
Yeah.
I mean,
Do I need to put it in the thing?
yes, yeah, you can grab one of these trays.
One drop, right? Or how many?
Yeah. Yeah, just a little bit, because we're gonna put one drop of the serum onto it, so it doesn't need a whole lot. And I did this slightly out of order, so I'm like trying not to make a mess of me.
Online folks, y'all still doing okay? So I guess y'all can look here and can I? There we go. So that's all we're doing. Looking for some blood on here. $111 is what mine is. So it's gone down a little bit.
Mine was 104.
104.
Mine was 129.
All right, so we had a couple go down, one go up. You had more, you ate two though, right?
Yes.
I think we just had one. So we are active enough that we burned through some of ours a little bit. So there you go. All right. Plus. I've got some toothpicks for you to use to stir those. Use a different stir stick for each well so that we don't contaminate. You may also want to do this on some paper towel. So that you can see the results.
And then we put the
Yep.
maybe put it in the B.
Yep, you're gonna put one drop of that into the B one. Very nice. Yep, 121 and 96. There you go. Eat some chocolate and then do it again. Like, what's your excuse? I just want to see it go up. That's all.
Okay, so what are the sticks are for?
They're just to stir it. So once like you're gonna put a drop of this in
Okay.
each one. So make sure you read the right one. You put the right dropper in the right one. So like this clear one, for instance, is the RH. So you're gonna put one drop of that, and then you're going to put a drop. You had one. And then you start with the station. Huh, that's interesting. Oh, it like tore. I didn't go in straight. That's why. It's like, why does this one hurt more? And then I can see like it's at an angle because I'm dumb. So like I said, here we're just looking for clotting. So if the liquid gets super cloudy and you don't see like clumps of cells, that means we don't have clotting. If instead we see some clumpage and the liquid gets kind of clear, that means we do have clotting. So you can look at mine and you can see some clots in the A. So see how clear the A is, and you can see some noticeable clots like that's right there. And there, you have some clotting there. So we have A positive. See how cloudy that one is? So that's not cloudy, it's just a bunch of cells. But so there we go. We have B. So A positive, A positive. That's we're negative from the edge. Because that's what we're doing here, is we're looking for the antigens. If we have the antigen, it will clot. So we have the A antigen present because we have clotting. We have clotting here, which means we have the RH side and A positive. There we go. It can be tough. You look O negative. Like I don't see any clotting in it. It's tough. So I'm gonna push it off the sides.
Yeah, because my family always knew I was O something. They didn't know.
This, I don't see any clotting in any of them. And usually especially with this particular test, usually the easiest ones to see is the O. And that is, yeah, there's some clotting in the O.
O positive?
Yep. See how I'm kind of tilting you now, you can see those little tiny dots.
The dots are
The dots are the clots. Yep. So O positive. There we go.
Oh, yeah. And if you
Don't let Carter Bloodcare know. Because you will get a tremendous number of phone calls. That one is not super common. And because it's not super common, they're always looking for it. It's also easy for them to run out of it. So about the only two things that get called more than O positive are O negative, because they just use it for anything and everything. So like O negative blood, they'll just give away like candy.
Yeah.
It's like, oh, there's a trauma and this person's shot. We don't know what their blood type is. O negative. So they go through O negative like crazy. So O negative gets a boatload of calls, not just because it's rare, because it's actually not that rare. It's just they use so much of it that they go through it very, very quickly. But AB negative gets a bunch of calls as well. So yeah, AB negative, O positive, and O negative, just they want you to come in literally as often as you possibly can. So by all means, do your good citizen thing and give them their, like, donate some blood. Just give them a fake phone number, unless you just want to get bothered all the time.
like that you're just phone numbers.
Yeah, exactly. I see, that's my kind of vindictiveness right there.
Here, put it under on some paper towel. So we got some clotting going on in there.
Oh, okay.
And some clotting in the A. B looks clean, but you can see some dots in RH and the A. So you're also A positive.
Yeah, let's go.
Now I know who to trap if I'm ever like bleeding profusely. Now, the last thing that we can talk about then is, while y'all are admiring that stuff, is the other things that are shoved into that blood, which makes up that less than 1% portion that we were talking about before. And that is to say, our leukocytes.
So our white blood cells. We have several different kinds of white blood cells, right? The two major groupings of white blood cells are going to be our granulocytes and our agranulocytes. So anything with blood doesn't need to go in that one. Granulocytes are going to have these little dots in their cytoplasms, okay? So we're going to see these dots, and these are basically going to tell us that we have like little vacuoles in here that are filled with something, okay? We have three types of those. We have neutrophils, eosinophils, and basophils. So neutrophils are going to be the ones over here on the left. So the top left one. They are neutral or neutrophils because their granules don't really stain very well. The granules are pale to clearish, but they are still present. Okay. These are going to be our most common type of white blood cell. These are going to be found in roughly 40% of your white blood cells are going to be these neutrophils, or even 45%. Next, we've got our eosinophils. That's going to be these top ones there with those red dots. So eos means sun, like a sunrise or a sunset. The suns kind of get that kind of reddish orange glow. That's what these granules kind of look like. They have this kind of red, orange, or reddish paint color. There's a bunch of them. Eosinophils are going to be anywhere from 1 to 4% of your white blood cell count. So they are quite rare. The second most rare, I guess. Not super common. That's because these have one very specific purpose. And that very specific purpose for our eosinophils is fighting multicellular parasites like worms. If that number of eosinophils is higher than 1 to 4%, that usually means that we have some sort of chronic parasite infection. So we have some type of worm infection. And that's why we have an elevated eosinophil count. Okay. Now, some people will have particular types of allergy, like types of allergic reactions or just kind of seasonal allergies due to having too many overactive eosinophils. So they will also have some elevated eosinophil numbers too. Then we've got basophils. Basophils have a whole bunch of really, really dark granules. So that's the one on the right top. So lots and lots of big purple dots. They're filled with these things. These granules in basophils, these make up anywhere from 0.5% to 1%. So very, very rare. These are filled with heparin and hold on, what's the other one? Heparin, and I'm having a brain malfunction. It's a vasoconstrictor or vasodilator. These cause inflammation, okay? Whenever you make a tissue angry by, I don't know, stabbing it with a little tiny spring-loaded lancet, it's going to cause the release of these basophils to these areas, and they're going to start releasing their contents to make your blood vessels dilate so that we can get more blood to them. And then we're also going to release a bunch of heparin to make sure that we don't clot that extra blood that's going through. Why? Well, it's so that we get more blood recruitment to these areas so that we can get white blood cells here to take care of any potential pathogens. But also so that we can get red blood cells and more nutrients and stuff to there so that we can repair those areas more quickly. That is why we have localized inflammation in these kinds of places, okay? Is so that we can speed up the healing process. Now, we also have agranulocytes. That's going to be these bottom two. So the one on the left is the lymphocyte. You'll notice that we have a very, very large, very dark nucleus that's very much round in the lymphocytes. Monocytes are also awesome. Notice that monocytes are huge. They're something like 20 or 30% bigger than all the other ones. These are super cool because this is their inactive form. This is their baby form, okay? These will migrate out of the blood vessels in a process called diapedesis, and then they will turn into monsters. They turn into macrophages. So they'll get even bigger outside of the blood vessels. This is as big as they can be and still fit through capillary beds, okay? They will leave the blood vessels in sites of injury or really just leave the blood vessels when necessary. And they will turn into macrophages, which will eat potential pathogens and dust, dirt, debris, bad cells, and all that kind of fun stuff. So they'll get even bigger than this whenever they turn into their active form, which makes them super cool. Now, like I said, all these numbers, histamine, that's the one I was looking for a second ago. I had, like I said, a brain fart. All of these things, though, have to be in these particular proportions, or will typically be in these proportions. If they are not in these average proportions that I'm giving you here, that likely tells us, one, that something is wrong, and two, the way those proportions are wrong will tell us what kind of thing is wrong. Oh, I didn't talk about lymphocytes very much. Lymphocytes are anywhere from 20 to 40% of our leukocytes. They have that big, dark staining round nucleus. We have three major categories. We have T lymphocytes, B lymphocytes, and then the best name ever, natural killer cells. T lymphocytes are going to turn on the rest of your immune response. B lymphocytes are what we use to make antibodies as well as have memory to diseases. Okay. So B lymphocytes are very, very important in our learned immune responses, as are T lymphocytes. But then natural killer cells. That may sound like, oh, ha, 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 funny, you called them that for no reason. No, that's literally what they do. Okay. They shoot holes into bad cells. They make a protein called aquaporin that literally punches holes in target cells and makes them shrivel up and die. That is why they are called natural killer cells, because that's how they work. They also fill them with a different enzyme. So they punch a hole in them with a protein called aquaporin, and then they have another set of enzymes called granzymes that go in and eat all the proteins inside the cell. So they literally punch a hole in them so water just gushes out the sides, and then they stick a protein in there that eats all the other proteins inside them. So calling them natural killer cells is not some like overly dramatic kind of thing that someone thought was funny. It's like, no, that's because these things are effing brutal. So yeah, they're super fun. We'll talk more about each of these things and all of the other white blood cells too once we get to the immune chapter, which is in a couple weeks. So next week, we're going to go over the heart. So next Wednesday, we'll actually do a heart dissection. We'll also do some EKGs of us so that you can kind of look at rates and kind of see some peaks and stuff like that. So that's what we're going to do next week. First day, it'll basically just be looking at parts and pieces, talking about how it works, look at some diagrams, look at some models. And then Wednesday, we'll look at a couple models again, then do a dissection and then do EKG. But the week after that, we'll start the immune system. I may even go get some rapid strep screens so we can do that as well. Oh, one of the other big things that comes with doing a differential count or one very common test that is ordered for blood when blood is drawn, especially in large amounts, is they'll do a CBC with a differential. Okay, that's how they'll order it. Okay, it means a complete blood count with a differential. That means we're going to be counting the number of red blood cells that we have so we can get a hematocrit number. But we can also get a count of all of the white blood cells. That's the complete blood count. And then we do the differential. The differential order is specifically so that we are going to count the number of white blood cells and their proportions so that we can see if they are in those correct proportions. If not, we can figure out kind of why that might be the case. Say we have too many neutrophils. Well, too many neutrophils means that we have some sort of bacterial infection going on. Potentially, anyway, some sort of bacterial infection. Stress can also do it as well. Neutropenia, which means we don't have enough neutrophils. Well, that's a problem. That usually tells us that we have some sort of anemia occurring, or that we have knocked them out because of doing certain therapies in some cases. Like, for instance, if someone has had, not a transfusion, but say someone is going to be going in for like a transplant of something like that. They will go in, and part of the therapy for doing the transplant is going to be knocking out a bunch of these white blood cells so that we don't attack the transplanted organ. All right, we can have a bunch of monocytes. Why might we have a bunch of monocytes? Well, if we have a bunch of chronic inflammatory diseases, we might have a whole bunch of monocytes. So if you look at someone, they have a monocyte count that's doubled or triple what it's supposed to be. It's like, oh, we have some sort of chronic inflammatory issue. Someone like with rheumatoid arthritis may have elevated monocyte counts. Basophils do the same thing. We know that this is causing like heparin and histamine. Heparin and histamine are also well known as being problematic for people with allergies. Most seasonal allergies are caused by those two chemicals. So typically those folks are going to have elevated basophil numbers. Lymphocytes are also very important. Like I said, we're going to see a bunch of these if we have an active or chronic infection, stuff like that. And then eosinophils, like I said, are going to be really common if we have a particular type of parasite infection. So we'll talk more about those next time. So now that we've done all of the in-lab work here, instead of having you put your own blood on a microscope slide and look at it, you can do that if you want. We have all the stuff. Or you can do the virtual lab to do that part. Okay. But because y'all did this part, I will at the very least check y'all off on the blood typing one. You can still do it if you want. It's not going to hurt you. It's good practice. But I'll at least check you off on the blood typing one so you don't have to do that one. Y'all good? Everybody here good? All right. Well, in that case, then y'all are free to do what you need to do. No, those you will place them in the back. This classes. Yeah, after a little while, it'll all just clot. Like we start to get the fibrin, fibrinolysis, and all that, or like fibrinogen thing going on, and there, so it'll just all clot over time. Yeah. Actually, let me see if I can do it. I've seen some, like I said, some fun videos of like bodybuilder viscosity. Let's see if I can pull one up. Oh, I'm not finding the one I'm trying to see. So we can see the pull here. I don't know what they're going to say, so I'll record this.
My kitchen, and he said that he would drain my blood for me. So what we did is we stuck the IV needle in my vein and we just left the other side open and we just
This is just after giving his blood right now.
from too much blood loss, but I needed to get so much blood out.
Like this is just after giving his blood right now. I mean, it's chunky and it's the thick.
See how thick?
This part's already coagulated.
Your
It's a
blood,
ton
you
of
give
blood.
yourself
I mean,
500
let's see how
milligrams per
that works.
testosterone every 2 days.
That's how thick my blood was. Like most
That's
people's
blood.
blood will like
That's his blood.
thicken over time, but mine was like
And he's not even huge.
like you could pour it upside down and the blood was solid.
See?
That's how thick it was.
And you can tell it's fresh because it.
What's different?
you
about
can
me
still-
than
you can tell it's
people
fresh
in
because it's still
Israel is
frothy.
that I prioritize health.
Oh, man.
Like, just don't. Like, who cares? Yeah. So, yeah.
Like, how can they, like, blow through your veins?
Yeah.
It barely does. Like that's why they will always have like elevated blood pressures and all that kind of stuff. And usually, like a lot of them will have to take like part of the course of them taking so much testosterone and stuff will also usually be some amount of anti-blood pressure drugs, like just to help keep their blood pressures down. Just as like we're going to give you this, you have to take this other stuff too. So you have to take this other drug to be allowed to take this drug. Yeah, basically. Or beta blockers and stuff like that.
Let's see.
I don't think they can donor their blood.
No.
How
Nope,
thick?
you wouldn't know. Like it would have so much other stuff in it too. But part of what makes those professional bodybuilders and stuff like that, one of the things that allows some of them to get to that professional level and stuff like that is that they are better at tolerating the drugs than a lot of other people. Like you've probably heard the term roid rage before. Like that amount of those chemicals like genuinely like changes mindsets and how they respond to things and how they see things. Like there have even been clinical studies on like conversations that they've had with people, and they have so many drugs in their system that they are literally hearing different things than the person is actively saying to them. Like when they repeat back what they heard, it's something actively different from what they were being told just because they saw it in the most negative, aggressive possible way. Um, like crazy stuff. So yeah, like roid rage is definitely like an actual thing. But the people that are really, really good bodybuilders are more able to handle that amount of drug without causing like big, crazy behavioral changes. Like one of the most famous bodybuilders ever is this dude named Ronnie Coleman. Uh,
Yeah. The Hulk, right?
uh,
That who acted the Hulk?